Today we're talking with Alex Mohr, who is a scientist. He has his doctorate in, is it nutrition, or what is your doctorate in exactly?
Nutritional sciences.
Yeah. And you've done a lot of research on fasting and micronutrients and how these have an effect on our body, and more specifically on our systems biology, specifically the microbiome. So over the past couple of decades, we've had, in the United States anyway, a pretty large view around the macronutrients that we're taking in. So what kind of carbohydrates are we taking in, proteins, fats, and it's been very much so about energy in, energy out in terms of weight loss and overall health. But in some of the studies that you've been doing, in your most recent paper in Nature Communications, you've been unearthing how time of eating and the micronutrients are actually having a really big impact on our health. Can you unpack what you found in this most recent study a little bit?
Absolutely. As everyone knows that's at all interested in health, and if you're interested in health, you're going to be interested in nutrition because everybody eats, and we all intuitively understand nutrition is really primary when we talk about trying to improve our health state, and also, maybe make some choices that we know are not so good. It's a very contentious space. Things can get very heated with people that I would consider more lay, all the way to the academy, where we see, as you kind of alluded to, calories in versus calories out, diet quality. Things can get very contentious. I like to take the view of looking at this kind of holistically. So you can't get too wedded to any one area of research or any one area of focus. We know that everyone does eat. Macronutrients are important. Quality of those macronutrients are important, as is when you're taking in those nutrients. Now, intermittent fasting, of course, has become very popular. I think a lot of people are starting to understand just how powerful it can be, particularly when we look at things that are pretty tangible to most people. If we look at clinical outcomes like weight loss, fat loss, even simple things like improving blood glucose, triglycerides, etc. But kind of in the spirit of what we're doing here at Theriome, we really wanted to take a deep dive into some of these more newer forms of data. The microbiome, we know, is tightly interconnected with our health, and the metabolome, looking at metabolites that we can detect in our blood or other sample types like even the feces, are really kind of revealing some very important latent factors in terms of improving our overall health state. So I'll kind of unpack this particular study. I will preface it by saying we're gonna get a little deep into the science. Hang on, we'll pull out sometimes, and we'll get a larger picture. So this kind of all makes sense. But these types of studies are going to be really fundamental and kind of changing our perspective on how we should view nutrition and how we should pursue nutrition for our own personalized health journey.
Yeah, and as I was telling you before we started this video, is I posted something that I thought was relatively benign onto a platform called Threads, which I was just playing around with it. It's kind of like Twitter, and I haven't really done much with it, but it's this new platform, and I just posted a couple things to see if it's something that I'd be interested in. I quickly found out that I'm not at all interested in it, because what I posted was, hey, if we eliminate added processed sugars from our diet, and we eliminate seed oils from our diet, and we eliminate things with high toxin exposure, such as glyphosate, from our diet, by default, you're going to eat a relatively healthy diet. It's not going to necessarily be perfect. There's always loopholes, etc. But those three things are very commonly found in high processed foods that are not good for you. So by eliminating those from your diet, and again, I'm not a nutritionist, so I was just kind of saying this, my own personal perspective. It's something that I did for a few weeks in July, where I just totally cut these things out. And I was like, wow, this is actually pretty good. But the response that I got is I got a bunch of registered dietitians responding to me with misinformation. This is false, blah, blah, blah. And I was like, man, there's kind of a lot of shills in this industry that are working for essentially the industrialized food. That really, really surprised me, though. So I think it's really important because you have this great body of information, and that's essentially putting out, saying like, hey, if you don't agree with us on like these nutrients and this food quality, these macronutrients, you are wrong. And this is like kind of the mainstream line of thought from what I gathered. You know, I could be wrong, but I think you are bringing a more nuanced approach to this, also through a great burden of science. You're doing rigorous science to better understand this. So I think it's pretty important because the way that we're perceiving nutrition is not just progressing, but it's actually becoming polarized a little bit from what I gathered. And so it'll be interesting to see what you unpack today.
Well, you know, to your comment about the great source of studies that we have, the great source of data, I actually think that is in many ways, that's causing the problem because we have so much to pull from. You can basically craft virtually any narrative you want about what the most optimal diet is going to be. I always try to operate from first principles. And, you know, being a scientist, obviously, we're going to get very deep, very nuanced in some areas, but we always have to pull back and understand the greater picture, and also understand the types of individuals that we're going to be dealing with and how we're going to be disseminating information and what is going to be most pertinent to really shape that person's health. There isn't a one-size-fits-all approach. I think in many ways when we look at, say, dietary guidelines that are presented and supposed to be representative of how a population should eat, we know that that's just simply not the case when we talk about different health states. You know, maybe for an individual that is morbidly obese, insulin resistant, that type of approach may not really work well for them. And as your health shifts, those recommendations might shift. Now, I think really the challenge with taking in and digesting all this data, again, keeping it in the frame of the bigger picture, is how can we integrate everything to kind of get, to make sense of all this and get a handle and a cohesive understanding. And a lot of that comes down to the advancements that we've made in systems biology. So systems biology, of course, we're talking about things like your genome. We're talking about, obviously, the transcriptome, where we're going in, we're looking at, you know, this at the cellular level, the machinery that comes into play when we're building proteins, their functionality, and then ultimately the types of metabolites that we're producing. I mean, this is how our body operates. And if we're pulling in all that really powerful data and transposing that against different health states, different interventions like we did with this particular study where we're looking at intermittent fasting, we get a very, very good nuanced perspective of what actually can be effective. And one thing I will highlight in this study is not everyone responds the same to these interventions. In fact, we have, and this is well noted in the literature, we have to certain diets, to certain even drug therapies, we have what we call responders and non-responders. So how are you navigating protocols for those individuals? And you know, this kind of opens the door for those types of questions. And I think, and I'm a believer in personalized health protocols and personalized profilings in terms of diagnostics. Like, what are you getting in terms of your laboratory tests? What are you testing? All that comes into play with really getting someone on the right path to taking charge of their health.
So you're viewing this data through the lens of systems biology and understanding the impact of nutrition on the microbiome, on the transcriptome, genome, well, not genome, but metabolome, etc. So you're looking at those different factors, and that's kind of the lens that you're using. What would you say I know it's hard probably to categorize the mainstream line of thought out there, but what would you say is kind of the lens that the mainstream line of thought in nutrition is utilizing?
Well, they're utilizing pretty well-established biomarkers. You know, obviously, a lot of the epidemiology focuses on things like body mass index, which that's kind of shifting a little bit. I mean, that can still be a tool to be used at a very gross level, but when we look at things like body composition, we know that's not taking into account how much muscle mass someone may have, where that body fat is displaced. For instance, you can have, say, a lot of subcutaneous fat, but that's not going to be as detrimental to your health as putting a lot of fat ectopically, or a lot of visceral fat that's surrounding our organs and almost acting as its own endocrine gland, pushing out a lot of pro-inflammatory cytokines and really perturbing our health. Others, of course, we know a lot. I mean, you can buy these blood glucose monitors, which I know the technology is really advanced, where we're now able to monitor that continuously, and I think those are relatively inexpensive for most people that are interested in health. And also your standard clinical markers. You can go to your doctor and get any regular metabolic panel that's including things like glucose. You can get triglycerides, all that kind of stuff. But the unfortunate issue with that type of data, at least when you're not pushing it through a bioinformatic pipeline, computers are going to be able to computationally, or leveraging things like artificial intelligence, they're going to be able to connect dots a lot faster than the human mind is. So if you're hanging your hat on a select few biomarkers, that may be fine at a gross level, but really when you're talking about at a person level, you kind of have to get a little bit deeper into the weeds. And when we start talking about profiling the metabolome, where we might have hundreds to thousands of metabolites, or the microbiome, where we may have thousands of species, and we're trying to understand, what's changing over an intervention, and what microbes are interacting with what metabolites, what microbes are interacting with what other microbes. That gets so complex. The human mind really has, not that we're not stupid necessarily, but the advancements that we've had over the last several years with things like machine learning, I mean, that computational power is able to make those connections, and then that can inform us, where we can condense a lot of that information into digestible modules or components, and then it makes the data very, very valuable, and we can look at the data holistically. Again, not hanging our hat on a select few biomarkers, which unfortunately the nutrition space has been doing and continues to do. Not everybody, but a lot of researchers, and of course the lay people are getting this information kind of secondhanded. So in this particular study, we took a broad survey of what was going on in the gut microbiome.
Quick. So you are essentially, you said you're assessing it holistically, which means, hey, I'm going out and grabbing thousands of biomarkers rather than 2, 3, 4, or 5. So the factor, you have hundreds of times more information that you're pulling than a typical study might in this space. And from that, you're saying we can develop a more nuanced approach and get a better understanding of what food is doing inside the body, rather than just kind of looking at these few biomarkers, which might be help when you're looking at general population. But there is the bio-individuality that needs to be taken into account, and this holistic vantage point where we're looking at thousands of biomarkers spanning the microbiome, the metabolome, the transcriptome, are really giving us an insightful view into what's happening.
Absolutely, and that's very well stated. So just briefly, for those that don't fully understand what the microbiome is and really how it's tied to our health, obviously, when we look at the gastrointestinal tract, it's not sterile. We actually have microbes, whether they're bacteria, whether they're fungi, archaea, even viruses. This area of this territory of our body is populated, and in terms of the actual biomass, if we were to extract out all those microbes and remove all the digesta and water and everything, we're actually looking at about 200 grams for the average person. So I always like to show this cupped hand image because you could effectively put that amount into your hands, right? So you kind of quantifies it visually. That small amount of biomass, when we compare it to our own body, is on par or greater than our own genetic content. So that's when we're profiling these microbes, we actually look at their genome, and that's why we call this, say, metagenomics when we're doing these types of analyses. And they're not just residing in our gastrointestinal tract doing nothing. As I show here, they have a lot of different functionality, from harvesting food energy, improving gut function, reinforcing the gut barrier, which we know is really important in terms of our immune system, and quelling inflammation. And they also have a really large impact on the metabolites that we actually see present in our body. In fact, it's estimated that approximately half of the metabolites in our body are synthesized or modified by these microbes in some way. So that being said, we know that these microbes have a profound impact on our health. Now, because they are located in the gastrointestinal tract, where we're taking in food, we're breaking it down, we're absorbing it, they're going to have a huge interaction with nutrition. So nutrition modulates the microbiome modulates our metabolism. So we see these three components being very tightly connected and very, very important when we're trying to understand health states. So this paper was published in Nature Communications. This was a pretty lengthy study in terms of going through all the data, the amount of data that we collected. And of course, we had some excellent authors. I led this investigation, at least on the analytical side. Dr. Paul Arciero led the clinical side of this, who has decades of experience in looking at intermittent fasting, has really laid the groundwork in better understanding intermittent fasting, and actually modifying different protocols to optimize various health outcomes that we'll discuss in a moment. We also had some excellent authors from our team here at Theriome that were fundamental in profiling the metabolome, such as Dr. Panish Jasby, Dr. Corey Wisner, and others. So this study was a randomized controlled trial. So this is when we're looking at dietary studies, at least now, this is kind of viewed as the gold standard. I think this is going to change a little bit as we are able to better leverage things like AI, particularly when we can do N of 1 trials looking at individuals over a period of time. We're able to make a lot of different collections. So this is kind of a standardized cut and dry study design. We didn't have hundreds of measures in terms of time points, but this is viewed as a very valid study design, where we had a run-in period for a week, meaning that individuals were following their normal diet, being monitored, making sure they weren't deviating. Then we collected baseline samples at what we call week 0 baseline. We had a midpoint assessment at week 4, and then we had a follow-up assessment at the end of the study at week 8. So here our population was 41 individuals that were overweight or obese, and they were randomized to one of two dietary regimens, one being an intermittent fasting diet with additional protein paced throughout the day. I'll explain that in a second. And then the other, which you can kind of consider our comparator group or our so-called control group, following a continuous calorie restriction type diet, adhering to USDA guidelines for Americans. So basically what is viewed as, at least from an institutional standpoint, an optimal type diet. So the reason...
And do you get into how many, by how much they were doing calorie restriction?
Absolutely, yep.
Yeah. And
that is, I was just going to mention, and that was actually one of the limiters on the time for this study, because this is admittedly, a shorter study. 8 weeks is not a long time. However, we really wanted to have a pretty stressful dietary event on these individuals to see the metabolic and microbiomic response. So we were actually limiting calories by 40% from baseline levels. So that's a lot. To quantify that in absolute terms, we were cutting about 1,000 calories off of these individuals' baseline diet. So you can think about that in terms of what you're normally eating throughout the day, continuously cutting calories by about 1,000 a day. That's to maintain that for 8 weeks can for a lot of these participants can become very problematic. We get issues like dropout, adherence can be questionable. So we were limited in terms of our timeline because of that. However, these individuals were tracked by they were tracked by a registered dietitian for that time period, and adherence was very, very high, above 90%, which is actually very good for these types of studies. So that's an important factor to consider. Now, in terms of the actual breakdown of calories, we see in that top box plot, looking at the difference between groups, there was no significant difference. So we were matching overall energy intake for these individuals. Now, because one group was intermittent fasting, we couldn't do that at a day-by-day level. This was equated over a week. So the individuals following the intermittent fasting diet were doing so 2 days per week, and then having their normal meal set up 5 days per week, where they were consuming more protein. And we actually, if you go look at the box plots there at the bottom, we see the distribution of macronutrients at each time point. And because this is a nutrition study and you are manipulating nutrients, particularly with the intermittent fasting group, we were having them consume more protein. We do see a difference, obviously, in the amount of protein that they were consuming at weeks 4 and week 8 in comparison to the calorie restricted group. And that was at the cost of carbohydrates. So they were actually consuming less carbohydrates in comparison to the calorie restricted group and more protein. Okay. So in terms of actual body weight loss, so a really gross metric from an anthropometric perspective, we see that from initial body weight, the intermittent fasting group lost around 8% of their body weight, which in clinical terms, we consider 5% to be clinically significant. And in an 8-week time span, that is pretty remarkable. In comparison, we look at the calorie restricted group. They also lost a substantial amount of weight, coming in around 4.5%, but not to the degree or the significance of the intermittent fasting group. So already we see this dietary setup. Again, there was a difference in protein-carbohydrate ratios, but equated for energy, this elicited a lot more substantial overall weight loss in those individuals. Now, when we assess the microbiome, there's kind of certain steps you want to take before getting right down at the taxonomic and functional level. So for this, we know that the microbiome, it's its own ecosystem. It has its own ecological properties. It really wants to maintain homeostasis, just like our body. It has a degree of resilience. So because of the amount of caloric restriction that we were eliciting in the study, we were very interested in seeing, okay, was there changes in the overall biomass of the microbiota? For instance, was there die-off because there just wasn't as much energy coming in? Was there change in the diversity of microbes that we see present, being the different types of microbes, their ratios to each other? One could surmise, based off of what we were changing from a dietary perspective, that we would see decreases in biomass and we would see decreases in diversity. And actually, that was the exact opposite. So we found that biomass actually slightly increased, as did diversity. And this was actually across both groups. But when we look at the actual community composition, what we found in the intermittent fasting individuals is this had a more pronounced shifting response, meaning that their microbiome shifted a lot more in terms of community state in comparison to those that were calorie restricted. And very interestingly, when we look at the bottom panel looking at specific microbes, we found this was largely driven by three main microbes at the phylogenetic level of the family, which normally when we do dietary studies and we're looking at the microbiome, we see things occurring at, say, the genus or the species level.
Can I interject real quick? So I have a question, and it's more of a postulation that I'm assuming you'd have to make, but do you think that the increase in biomass with calorie restriction and intermittent fasting is because our body is, to a degree, going through a stress, and so it's increasing the amount of microbes so that we can have better nutrient harvesting?
That's actually a great interjection, Micah. And again, I would have to speculate, but I am thinking along those lines, almost a stress response,
like
offering a
exercise,
buffer, right? Offering a buffer. So we might see some hormesis occurring here, which I think kind of ties into the resilience aspect that we might see when we perturbate a system. So absolutely, that's what we could be observing. Now, very interestingly, when we looked at some of the key shifts at the family level that were occurring in those intermittent fasting, we saw some pretty notable microbes present. For instance, Cristinalaceae actually increased the most. And this is a microbe that's well regarded to represent or be present in someone that has a lean phenotype, meaning that they're metabolically healthy, they have lower body fat mass, they have greater muscle mass. We also saw other microbes like Rickenolaceae, which has been linked to reductions in visceral adiposity and greater metabolic health profiles. Now, very interestingly, when we look at some of the clinical markers, again, the basic clinical markers, like we can do DEXA scans, which we did in this study. So that's really kind of considered the gold standard when we monitor someone's body composition, where we're able to actually use x-rays and compartmentalize different tissues. So we can look at things like lean body mass, we can look at subcutaneous fat, and we can do it by body part. We can also look at visceral fat. And for the individuals that were following the intermittent fasting diet, they actually lost 33% of their visceral adiposity, which was well and above what those following the calorie-restricted diet were doing. So here we kind of see
how much did calorie-restricted diet lose?
I believe they were a little bit over 10. So
yeah, that's the stuff you want to get rid of. That's the stuff that has very negative long-term effects.
Right. So here we see almost a doubling or tripling.
Yeah.
Very, very, very impressive. So that's another factor when we talk about intermittent fasting, because it's not solely about the nutrients. We're also kind of training the body on pulling from fat reserves. And one thing that we have noted with intermittent fasting research is it's very potent at pulling it from ectopic or visceral fat, which is really the fat that we want to target, or even liver fat is really the fat that we want to target first and foremost. So another interesting microbe that we saw increase in this group was Marvin bryantia, and this has been kind of lauded as a candidate marker for predicting long-term weight loss success, which we actually will go into with one individual that we tracked for an entire year from this study, and we found some pretty profound effects in terms of remodeling not only their microbiome but also their metabolome. Now, again, kind of pulling back, we can see shifts in microbes, and they are obviously tied to our health, but we're not going to immediately feel that. It's not like, oh wow, that my Cristinalaceae has really gone up. It feels so great. We also have to, with these studies and with any type of monitoring of a patient, for instance, we have to tie that to basic things like, how are you feeling? So we did monitor gastrointestinal symptom rating scores, which is a validated clinical panel that we normally use, particularly in the context of things like IBD, IBS, gastrointestinal type diseases. And we actually found that those that were intermittent fasting had a significant decrease in total symptom scores. So this is monitoring symptoms from not only the lower regions of your gastrointestinal tract, but also the upper regions of your gastrointestinal tract. So think of things like heartburn, burping, things like that. But a lot of this was actually driven by largely the symptoms that were coming from the colon. So, as uncomfortable as talking about some of these things are, most people in the U.S. suffer from things like constipation, gas, stomach rumbling, pain in that area. So that was pretty profound to see. And in the short course of this study, we were already seeing beneficial effects that people could immediately feel. So, that's.
And I'm sorry, but did you say the window in which the intermittent fasting group was eating, and then the caloric restriction, was that 3 meals a day, or?
Yeah, that's a good point. So when they were feeding, they were consuming 3 meals per day. They did have a high protein snack. And when they were doing the modified fasting protocol, normally when we think about fasting, we say, okay, you're going to fast for 24 hours, right? But really, in terms of consumption of your last meal and then initiation of your next meal, if you're going to do it for a full day, it really translates into 36 hours. Because say we stop eating at 8 p.m., the next day, fasting all day, when we initiate food again the day after, we're sleeping. So we're adding those additional hours. So these individuals were doing fasting for 36 and then past 60 hours when they're doing the 2-day back-to-back fasting days. So that's an important.
They were doing that continuously throughout this, or
correct? Yes.
Yep. Okay.
Yep.
Yeah, that's a long time to do that for 8 weeks.
Yeah,
kudos to these participants.
These clinical trials can be a real bear to conduct, and of course, that's why you have these long author lists, because you have a big support staff making sure that these are successful. So, because the microbiome is so intertwined with our health, one area that we often look at is inflammation, or as some people like to call, particularly when they're profiling a lot of cytokines, the inflammasome, because we can add an ohm to every single data set. You can have the metabolome, you can have the microbiome, you can have the proteome, etc. So, as scientists, we like to make things cute sometimes. But the gastrointestinal tract is very proximal to our immune system. In fact, we know that gastrointestinal associated lymphatic tissue, or GALT, is comprising about 70% of our immune system. So that really shows you just how important assessing the microbiome is. And if we're going to do that, it obviously makes sense to look at things like cytokines, which are inflammatory factors that can be produced from these tissues. So we surveyed a panel of 14 cytokines, and we noticed that the intermittent fasting protein pacing group actually significantly increased some pretty notable cytokines, the ones that I show you here, including IL-4, interleukin-4, IL-6, IL-8, IL-13. And these are notable, I'll just kind of break it down. For instance, IL-4 has been reported to really display lipolytic effects, meaning it helps break down fat. IL-8 is really associated with positive reductions in weight loss and weight maintenance. IL-6 is interesting because it's kind of reported as a pro-inflammatory myokine, actually myo coming from the muscle, but it can acutely increase lipid mobilization, particularly in that tissue, showing that we're releasing fat from not only adipose tissue, but also muscle tissue. So a lot of people when they think of storing different macronutrients in muscle, they think primarily of glycogen, storing carbohydrate. But if someone's in a metabolically compromised state, we can also start storing fat in other places that we don't want it. Like the liver is one, actually a very lean tissue, but also starting to deposit fat into muscle tissue. So a really good example, you may like Wagyu beef. It's very tasty. That's because a lot of the fat has been deposited in that tissue, but these cattle are not healthy, and we don't want to see that, obviously, in humans either, and that's tied to metabolic derangement. So seeing release from these tissues is pretty remarkable. And then IL-13, which is very important when we look at the immune system surrounding the gut, and is actually a stimulator of mucus production from goblet cells. So these are little specialized cells that reside in our gastrointestinal tract and help release mucus that actually surrounds the lumen. We do a cross-section of the gastrointestinal tract. You don't have microbes just butt up against your brush border cell. You actually have mucus there to kind of protect those cells and protect and kind of quell immune response. So here we're seeing some really beneficial shifts systemically in the body. These cytokines were measured from blood. And again, both groups were calorically restricting. However, it really seems that the change in the macronutrient distribution, and particularly the fasting, is a really powerful lever for modulating inflammation. Now, moving on, we also profiled the plasma metabolome. So when we talk about the metabolome, we're really talking about these smaller metabolites that are downstream, or really like the end of the systems biology pipeline. We're actually seeing expression and what the body's producing, what these microbes are producing. And again, here we saw a pretty differential response in these two different dietary regimens, particularly increases in acetylcarnitine, which we know is pretty consistent when we talk about fasting, because this really can tie into releasing fatty acids, fatty acid mobilization, and increased transport of fatty acids via carnitine acetylation into the mitochondria, actually burning this fat via fatty acid oxidation, which we want. We want to get rid of that, fat. So making your mitochondria work and become more efficient. And this is actually pretty consistent with being in a ketogenic state. Relatedly, as you see that really bright red box, that's malonic acid, and we see a pronounced increase pretty quickly with the study at week 4. And this is a naturally occurring organic acid, and it's really a key regulatory molecule in fatty acid synthesis via the conversion of acetoacetate, a ketone. So this is really showing again that we're mobilizing a lot of fat, which was reflected in the gross clinical markers that we detected with, say, things like loss in body fat, and as I mentioned prior, with the really pronounced reduction in visceral adiposity.
Yeah, this chart is almost the opposite of each other.
Yeah.
It's pretty wild.
Yeah, pretty wild. In terms of the different pathways that we saw, most notably, and this was actually seen in both groups, but glycine, serine, and threonine, that pathway has recently been found in preclinical models to play a pivotal role in longevity and life-sustaining mechanisms, kind of independent of diet. So, while we didn't profile everything and we only had those 3 time points, we're really seeing some very positive changes associated with actually both diets, but to a more stark degree with the intermittent fasting protocol. So I want to bring this up And this was actually, you can read the full study to get a better understanding and go way deeper than I'm going here. But again, all these comparisons that I just showed you, we're looking at a group level, okay? And of course, we know that this is normally how we do research. This is normally how treatments are applied in a clinical setting. We're looking at population norms. We're seeing, where you fall in that range. If you're deviating, then it's, some kind of course correction might be warranted. But what's going on with an individual? Because we know that not everyone is going to be responding the same, right? Everyone has their own unique genetic makeup. They have their own unique microbiome makeup as individual as something like the genome. So we're really interested in looking at what goes on long term. And due to feasibility studies, or due to the feasibility issues with studies like this, we weren't able to track everybody for a year. But there was one individual that was actually morbidly obese coming into this, and they were really motivated, and they really wanted to continue on with this intermittent fasting protocol. So during the 8-week study period, they actually lost 15% of their initial body weight, which is massive. It's actually translated into 25 kilograms. So you can do the math. I mean, that's a lot of weight. So this person obviously was coming from a position where they could lose a lot, and because of the amount of energy that they were carrying on their bodies, they could go through this type of regimen for a year and be fine. In fact, there is a case study, a published case study of a morbidly obese individual, a lot more obese than this, the guy that we followed in our study, but this case study from a guy from Scotland, he fasted completely for, I believe it was 2 years and lived. So your body is...
Yeah, I remember seeing that.
Yeah, your body is incredibly resilient. And it just goes to show you how much energy that you can store on your body. And of course, that is a protective mechanism that some people express a little bit more in terms of their genetic makeup, but it's a protective mechanism that we've developed over the eons for instances of, if we have periods of starvation, which was actually quite a normal occurrence. And I would also argue that fasting was a very normal occurrence, and that our physiology is kind of coded for that in many ways. And I'm speculating a little bit here, but to your point earlier at the beginning of this, Micah, when you were saying, well, it's kind of intuitive that we would eat unprocessed whole food. I mean, any layman would say, yeah, that's kind of how we've developed. And normally when we adopt those types of dietary patterns, a good example is if you go to a grocery store and if you shop the perimeter. I mean, everyone knows this, right? You shop the perimeter, you're going to, say, your produce section, you're going to, the meat. Eating that type of food, if you eliminate stuff in the center, a lot of the processed, refined products, you're probably going to get healthier. You're probably going to lose weight. I think the same can be said with periods of fasting, because in many ways, throughout our history, we had this in our life cycles, and we get closer to what our physiology is really in tune for. I think we can return to a healthier state a lot more rapidly. So anyway, this individual we followed for a year, and we were a lot more granular with our sampling period. So as you can see here, with the blue line, with those line charts to your right, we were tracking their body weight over the year. So I have that in weeks from week 0 to week 52. And they had, an initial weight loss period that really kind of peaked around the 12-week mark, where they lost. I think this individual ended up losing over 100, maybe 110, 120 pounds. And for the most part, kept that off and remained weight stable, which is remarkable if you consider the phenomenon of weight, basically yo-yoing. So a lot of people will go on diets, they'll be successful for a period of time, they'll lose weight, but then they'll actually end up reverting back over time to their original weight and actually regaining more. So it's almost like, why did you even do it in the first place? Because you actually ended up worse off than you were before. To see this maintenance is really, really important, really clinically important. And this is the type of scenario that we want to see. And again, going back to the issue of adherence that I was talking about, running these types of studies, you want to find a regimen where someone can stick to it. And for a lot of people, intermittent fasting is attractive over just straight calorie restriction because you are having periods or days where you might be eating a little bit more and then periods where you're restricting. From a mental standpoint, again, something that we didn't go too deep into the study, this was more metabolism focused, is a huge part of diet. We know that you can have the best diet in the world, but if you aren't going to be adherent, if you're having some emotional instability, it's just not going to work out. So this is the type of scenario that we want to see, this weight loss and then weight stabilization. And we compare that to what was going on in the microbiome. We saw some initial increases in diversity that actually tracked with the weight loss. And looking at the overlay of the intermittent fasting group, we actually see difference in response. So this variation we know is really, really important. As I was saying, we can have low responders to dietary intervention. We can have high responders to dietary intervention. So here we see in this bar chart to your left, this is the amount of weight loss that all the individuals that followed the intermittent fasting diet experienced over the 8 weeks. So some lost over 10% of their initial body weight, and some didn't actually break the 5% mark. So they were following, equated for body weight, energy intake, they were following effectively the same type of diet, but here we see a vastly different response in weight change. And this was reflected when we look at the microbiome. I won't get too deep into some of these complex volcano plots and charts, but basically their metabolome, their microbiome, we saw differential response, particularly the individuals that lost the most weight displayed enrichment with fecal metabolites involved in lipid metabolism, where those that didn't lose as much weight didn't have this type of response. In fact, they were more prominent in pathways related to extracting energy. So their microbiomes, again, going back to having the stress response, may have been under stress and we're like, look, we're getting starved. We need to take the foodstuffs that we are getting in and pull out as much possible energy as we can to, not only preserve our state, but also the host, because we know of that deep connection that we have.
So, and did you find a difference in their microbiome diversity going into this? Was there kind of a common link between the people that were in the low? And then the follow-up question would be, if you were hired by those 5 people to assist them in losing weight, would you do targeted interventions? Would you change up the diet to something different? How would you approach it? Absolutely.
So assessing baseline, it's always important to see what differences you see, what you see with obviously a group, if you're looking at study or a population. And these individuals did have differences in their overall microbiome composition. So the individuals that didn't lose as much weight actually had microbes that are basically good at producing energy, principally actually butyrate, which we know is a short-chain fatty acid that we can produce from the microbiome. There's other short-chain fatty acids that we can produce, but that's really the main driver in terms of extracting energy and actually giving energy back to the host. So we can actually absorb butyrate, and that can be a pretty substantial contributor to the overall energy that we're getting into the system. So these individuals already were showing some resistance to being in a deprived state. In some ways, they might have had a more resilient microbiome than those that ended up losing more weight. We're limited, obviously, in the sample size, but in terms of individual profiling, like when we start talking about at-home testing, for instance, understanding where you are coming in before any intervention is, in my mind, really where things are going to shift, because now you don't have to say, well, intermittent fasting, that's really popular right now. I guess I should do that.
Let's do it for 8 weeks and figure it out. Yeah.
Right. Really, I highly recommend you get a profiling beforehand. And there's a lot of companies now out there in this space. Of course, we have Theriome, which I am a firm believer in what we're doing. I think that would be a great route, but there's many others that you could go. Of course, there's great physicians out there, some that we've worked with that are really embracing this and really understanding that initial profiling, you're going to save so much time in terms of finding an effective protocol. And then that, of course, translates into adherence, because no one wants to go into something blind and not get very good progress. That's disheartening, and that ties into compliance with the regimen, that ties into adherence. And I think in a lot of ways, this yo-yoing that we see, because if people get good results right away without feeling like crap, again, tying into the motivational and emotional aspect of this, they're going to make rapid progress, they're going to feel good, and they're going to save a tremendous amount of time and really make good strides in improving their health.
Well, I think this is a good point to put in right now on the microbiome testing, but this study is showing a couple of things. One, it's showing the validity of intermittent fasting over your standard calorie restriction, because people were getting overall a much better response. Even the ones in lower weight loss seem to have a positive response in microbiome shift, and although it wasn't as stark as the ones that had a high weight loss, it's still positive, right? But on the flip side of that, you're applying the same protocol to all these people, and you're seeing pretty drastically different results across the board, considering that this is a pretty similar group of people. And so when you're a clinician and you're considering your patient base, do you have the person that's the 15% loser over 8 weeks, or do you have the person that's a 4% loser over 8 weeks? 4%, 15 loser probably isn't quite the best term, but loss in weight. But I think the point there is that profiling them to understand their trajectory prior to this happening is really important. And with the microbiome test so you do deep sequencing, right? You really get an understanding on what's happening in the microbiome. But there's the other component of this too, which is what do you do with this data? So how have you built in personalization into this test so that the clinician is equipped and they don't have to filter through line and line and line of data and data and data? They can get an understanding of what this person is going to respond to based on the information that you're providing. So can you perhaps go into a little bit what you've built into the report?
Yeah, and not to throw shade on other tests that are out there, but I think it's a real disservice, again, depending on what you're trying to do. If you're trying to do a very specific, for instance, pathogen or parasite panel, or you're interested in performing a probiotic panel, then yeah, you want to look at individual microbes and take some type of steps towards correcting or eliminating in the case of parasites and pathogens. But when in a report you get a breakdown of the microbes, well, that can be kind of interesting to see, what a microbiome is made of. You're still putting a lot of the burden on the clinician to connect the dots and figure out what we need to do. And a lot of that's just going to be drawn from the literature. Okay, what is this microbe associated with? And then you're going to take some shots in the dark in terms of, okay, I want to modulate this particular population of microbes, or I want to decrease, I want to decrease these metabolic pathways. That we all know that physicians are strapped for time, just like. Not that they don't understand all the data, It's just that, again, the human mind can only comprehend so much. So I think where we really shine is condensing all this information into modules. So again, we know that the microbiome is intricately tied to the immune system, for instance. So we have an immune module. We know that this is intricately tied to digestion and absorption of nutrients. So we have a nutrition module. So right off the bat, we can find the main nodes of connectivity when we look at this data, and then find what scenario is going to work best for shifting these, whether we're talking about clusters of microbes or the overall community, what we can really move the needle on in terms of the intervention that we might be recommending. So this kind of arms the physician with things that are actually going to work, and we even have like kind of a hierarchy of what is going to have the most effect, and then we kind of can go lower from there. For instance, I understand probiotics are popular. I understand that they can have some positive effects, but in terms of modulating the microbiome, we know that we can have more profound effects with things like, for instance, dietary fasting. And those protocols, of course, depending on what we're observing in the data, that's built into our algorithms, and those type of recommendations can be made, as well as dietary and other important environmental considerations. Even one that's not talked about very often is hydration, and even heat stress, which we know can affect the system. So condensing all this complex information into modules, using our digital twinning platform, where we're actually able to compare the person to them, basically to themselves. We're not making those population level inferences. We're seeing what works best for that person, and we can really create tailored, bespoke interventions or recommendations that are going to see some positive changes.
Yeah, and to kind of oversimplify this, your report, so in this study, right, you're taking a group of people and you're applying the same thing to them because you're trying to see what is happening when we apply the same thing to people.
Correct, yeah.
But when you're a clinician, you're really trying to assess what does this patient in front of me need. And so the report that you've compiled takes into account your understanding of biology, your understanding of the microbiome, and it's about taking, again, to oversimplify, those people on the left with that blue chart there, and they didn't have as much weight loss. How do we get them into the gray or possibly even into the yellow? And so you've built in these recommendations based on their biology that are going to shift them into whatever it might be: better weight loss, reduction of disease, well, I shouldn't say that, reduction of negative microbiota, etc. And so it really is an individualized approach using a holistic vantage point in our understanding of biology, and then kicking out these very actionable steps that are going to bring people into a microbiome associated with a positive health state.
Yeah, and again, this is the way that studies are traditionally conducted, the randomized controlled trials. It's really what everyone views as the gold standard. As I was saying, really as we start realizing a lot of this technology, we can do these N of 1 studies. N of 1 studies is what physicians have been doing since forever, right? They're a case study. They're following an individual. They're getting, they're using various tests, and they're assessing when they deem appropriate. What we're trying to do is come in and take away a lot of that guesswork. So everyone's case is going to be different. And that even goes into when you need to do retesting. For instance, with this case study, and I'll advance the slide because I'll show you the shifts that we were seeing in the overall microbiome and really the restructuring of the microbiome. These were taken over a year-long period. At weeks 0, 4, 8, 12, 16, 32, 52. Kind of logically segmented, but that may not be the right sampling cadence depending on the person. So for instance, we see massive changes occurring between baseline and week 4. If we really want to get a better handle on someone's transition or this restructuring, this may not be the most optimal sampling cadence for them. So that's another important consideration when we talk about testing. And some people may require more, some people may require less. We really need to start viewing everyone as an individual, and that's the way that these tests need to be conducted. So yeah, in terms of the restructuring of the microbiome for the individual that lost the most weight, again, this is the best case scenario. The reason I'm showing it to you, obviously, is because we did track this individual, but it really shows you, I think, the potential for what can be achieved. Now, obviously, this is not going to be the starting point for a lot of people, but it really does show you how profound an optimized protocol. This person responded really, really, really well to intermittent fasting with greater amounts of protein in their diet. Everything from increasing certain bacteria that we know have a profound effect in mobilizing fat via what they're producing metabolically, to increasing mucus levels, particularly with an increase in Akkermansia mucinophilia. This is a special microbe that actually is kind of viewed as the lawnmower of the mucus that we have in our gut. Not that that's a bad thing necessarily, because we do want to have a higher turnover of mucus to keep that layer nice and healthy. So we see this transition from a disease associated microbiome to a health associated microbiome, and those changes were happening very, very rapidly, even at week 4 out to obviously the end of the year. And this was really kind of, there was some changes, but this was really kind of maintained. And this is what we want to see. Obviously, if you're a physician, we want to see this normalization, this movement back to health, and we want to see that those outcomes stabilize as the person's health improves. So overall, this kind of, I think hopefully, some of this might have been intuitive, but I hopefully this kind of changes your understanding of nutrition a little bit in the context of the microbiome, because we get so caught up in what the perfect diet is, whether we're looking at ratios of macronutrients like protein, carbohydrate, fat, what type of foods we're taking, the quality. While all that's important, we understand, and I don't want to say it's just as important, but it's certainly very, very important. We know that when you're eating and when you're fasting also has a profound effect on this environment, which directly translates to the interaction with our body, and whether that's tied to mobilizing fat tissue, modulating inflammation, or just making us more metabolically efficient, having our down to things like our mitochondria just working more efficiently, because now they're chewing through all that all those fatty acids, producing a lot of ATP, a lot of energy. I know, again, intermittent fasting is not going to work for everybody, but when you're in a certain compromised state, and if it is kind of in line with what's going to work for you, ultimately we want people to just not only be healthier but also feel better, I think, which ties into a lot of what we do on a day-to-day basis that kind of dictates our choices. So that's the study, and I really appreciate going over it with you, Micah.
Yeah, thank you. It's pretty fascinating to be able to see, and we kind of seeded the fact that there's the microbiome test. So for anybody's interest that piqued, it is not yet available to all doctors and the public. We anticipate in December that Alex's microbiome test will be available to clinicians. Right now, though, what we are doing is we're doing a beta test. So we've done the research on our side to understand this test, the report, etc. And so we're working with 5 practitioners that are going to be running 10 tests each. Currently, we have a few, but we have, I believe, 2 slots open where practitioners can join us if they want to purchase 10 of these test kits and try it out with their patients. They get early access to this, and then also conversations with Alex and honing this in a bit more so that we can make it more applicable to the clinicians. And it's less on the science side and more of like, how is this being communicated to you? How can we make the the design, the communication of this better, etc. But that's the stage we're in with the microbiome test. So if you are a clinician and you are interested in trialing this with 10 patients, then you can send an email to me, micah@therio.me. I'll put it on the screen here, and we'll get you into a conversation with Alex and go from there. But this is, again, it's for a limited time. We're starting in a few weeks here. We're going to be working with 5 practitioners, 10 test kits each, so there's limited space available. If that is something that you think could be impactful for your patient base and better understanding them, then please do contact us. And you can see our website there, www.therio.me, and we'll get everything handled that we can. So appreciate your time, appreciate you watching, and thanks, everybody.